Let me guess, answering a biology paper, yet got stuck in a certain question? Well, glad you clicked on this video. Welcome back to my channel, where students of biology are simply interested in learning more. Today, I will be going through the 9700 May-June 2020 Paper 12. If you are new to this channel, then do like and subscribe, else just continue watching. Question 1. What is the definition of the resolution of light microscope? The keyword here is the definition of resolution. Resolution is the ability to distinguish between two objects as separate when viewed by a microscope. Therefore, when going through these options, A. The degree of sharpness produced by the microscope. Well, that doesn't define resolution. The greatest distance between two objects visible in the same field of view. That is also not the correct definition. The minimum distance that allows two objects to be viewed as separate. Now that is the correct answer. Therefore, the answer is C. Question 2. The electron micrograph shows a type of virus at a magnification of times 30,000. What is the length of the virus? Now, magnification is nothing but the image size upon the actual size. They have given the magnification as 30,000. Therefore, when we substitute the value 30,000 into magnification and we find out the length of this virus given in the image, which is the image size, we get 66 into 10 to the power 6 nanometers. Hence, we solve for A, which is the actual size, we get 2.2 into 10 to the power 3 nanometers. Therefore, the answer is A. Question 3. Which row correctly shows a feature of a cell structure? They have given the two columns as site of protein synthesis and makes lysosomes. Protein synthesis occurs in ribosomes and in the RER, which stands for rough endoplasmic reticulum, whereas lysosomes are made in the Golgi body. Therefore, if we go through these options, site of protein synthesis is ribosomes and makes lysosomes is Golgi body. Therefore, the answer is B. Question 4. 4 students were asked to match the function with the appearance of some cell structures in an animal cell. The functions were listed by a number. 1. Organizes microtubules to produce a spindle during cell division. 2. Synthesis of polypeptides. 3. Packaging of hydrolytic enzymes that will remain in the cell. The appearances were listed by a letter. V. Membranes which surround an enclosed inner cavity. W. Non-membrane bound spherical structures. X. A double membrane with many pores. Y. Non-membrane bound cylindrical structures. Z. Membrane bound sacs arranged as a flattened stack. Which student correctly matched a numbered function with the appearance of the cell structure? Now in this question, we have to match the function with the correct appearance. So function 1 is nothing but centrioles, as it organizes microtubules to produce a spindle during cell division. Function 2 is ribosomes, and function 3 is the Golgi body, as the Golgi body produces lysosomes. Therefore, if we match the correct appearance, 1 would be non-membrane bound cylindrical structures, 2 would be non-membrane bound spherical structures, which is letter W, 3 would be membrane bound sacs arranged as a flattened stack, which is Z. Therefore, the answer is C. Question 5. The features of some cells and cell structures that make ATP are listed. 1. Has outer boundary membrane and folded inner membrane. 2. Has peptidoglycan cell wall outside and outer boundary membrane. 3. Has a double boundary membrane and stacks of inner membranes. Which row identifies these components? So I have given the structures with double membrane. Structures which have double membrane are the nucleus, mitochondria and chloroplast. The structure which has a folded inner membrane is the mitochondria, and the structure with stacks of inner membranes is the chloroplast. Therefore, the correct answer is D. And obviously, bacterium has a peptidoglycan cell wall. Hence, 1 is mitochondria, 2 is bacterium, 3 is chloroplast. Question 6. Mitochondria are thought to have evolved from prokaryotic cells that were ingested by an ancestral cell. Which feature have these prokaryotes lost during the evolution into mitochondria? Obviously, mitochondria do not have a cell wall. Hence, the answer is A. The feature which the prokaryotes have lost during the evolution is the cell wall, as mitochondria have a mitochondrial envelope, which is a double membrane instead of a cell wall. Question 7. A student carried out the Benedict's test on a sample and got a negative result. That means there are no reducing sugars. What should the student do to confirm there are no sugars present in the sample? The student should carry out the non-reducing sugar test, and the non-reducing sugar test is done by boiling the substance with hydrochloric acid, neutralizing it with alkali, and repeating the Benedict's test. Therefore, the answer is C. Boil with hydrochloric acid, neutralize with alkali, and repeat the Benedict's test. Question 8. The diagram shows 3 hexoses. Hexoses means 6 carbon sugar units. In which carbohydrates do these 3 hexoses occur? So structure 1 is alpha glucose, structure 2 is fructose, structure 3 is beta glucose. Now how do we know whether it's beta or alpha? Alpha has the OH group below its plane, whereas beta has the OH group above its plane. Therefore, in 3, the OH group lies above its plane, therefore it is beta. And in 1, the OH group lies below the plane, therefore it is alpha glucose. Sucrose is made up of glucose and fructose, and since option 2 is fructose, it is C because 2 is fructose and fructose belongs to sucrose. Cellulose is made up of beta glucose, therefore 3 belongs to cellulose, and 1 is nothing but alpha glucose, and alpha glucose makes up glycogen, therefore 1 should belong to glycogen. Hence the answer is C. Question 9. The diagram shows part of the chemical structure of a naturally occurring polysaccharide. What types of glycosidic bonds are present? As you can see in this structure, the neighboring units are linked at 180 degrees relative to each other, hence it is beta instead of alpha, as you can see a rotation. Therefore, C and D is beta glucose, and if we look at the linkages, we have to count which carbons are joined to which neighboring carbon atom. Therefore, if you number the carbon atoms starting from 1, you can see that the linkages are beta 13 and beta 14. Therefore, the answer is C. Question 10. What is the maximum number of water molecules produced when a triglyceride is synthesized? A triglyceride consists of 3 ester bonds as it undergoes condensation reactions to form 3 water molecules. Hence, when a glycerol molecule joins with 3 fatty acids, 3 water molecules are produced. Therefore, the answer is 3, as 3 is the maximum number of water molecules produced when a triglyceride is synthesized. Hence, A is the correct answer. Question 11. What occurs during the formation of a peptide bond between 2 amino acids? In peptide bond formation, if we recall correctly, OH is removed from COOH and H is removed from NH2 between 2 adjacent amino acids. Therefore, the closest and correct answer is option B. OH is removed from COOH and H is removed from NH2. Question 12. The diagram represents roles of water in living organisms. Which roles are dependent upon hydrogen bonds between water molecules? So they have given the roles of water in living organisms. 1. Carries dissolved substances. 2. Provides a constant environment for aquatic organisms. 3. Temperature control due to evaporation. Hydrogen bonds affects the specific heat capacity. Hence, the roles 2 and 3 are dependent upon hydrogen bonds. Therefore, the answer is D, 2 and 3 only. Question 13. The diagram shows a metabolic pathway. What would be the effect of adding a small amount of a non-competitive inhibitor of enzyme 2? They are talking about a non-competitive inhibitor being added, but a small amount. Since a small amount is added, there should be some amount of enzyme uninhibited too. Therefore, option C is incorrect. Substance Y would no longer be formed is incorrect. However, substance X would increase as some are inhibited by enzyme 2. Not all are inhibited, but some. Therefore, substance X would increase in concentration. Hence, the answer is B. Question 14. 2 enzymes are added to a solution containing a low concentration of a substrate that they can both use. Which statement is correct? Enzyme with lower Vmax will have a lower Km value. Therefore, lower the Km, which is the Michaelis constant value, higher the affinity towards substrate. Hence, we'll use more than the other enzyme. Lower the Km value, higher the affinity towards the substrate. Higher the Km value, lower the affinity towards the substrate. Therefore, the answer is D. The enzyme with the lower Vmax will use more, as lower the Vmax, lower the Km value, lower the Km value, higher the affinity, more substrate will be used. Hence, the answer is D. The enzyme with the lower Vmax will use more of the substrate than the other enzyme. Question 15. The diagram shows the fluid mosaic model of membrane structure. Structures 1, 2, 3 and 4 are types of molecule. Which types of molecule are always identical? In a fluid mosaic model, only cholesterol will always be identical, as others act as antigens and cell signaling receptors, hence could be different in shape. Therefore, the answer is D, 4 only, as 4 represents cholesterol. Question 16. What is the role of G protein in cell signaling? Over here, the correct process done by the G protein is the release of a second messenger. Therefore, if we go through these options, closest and correct answer is B, to act as a switch releasing a second messenger. Question 17. When a small quantity of phospholipid is added to a test tube of water and then shaken vigorously, an emulsion is formed by small droplets called liposomes. Which diagram shows the arrangement of phospholipid molecules in a cross-section of a liposome? When shaken, the molecule will rearrange with polar heads being on the outside surface and non-polar fatty acid tails being protected from the watery surroundings. Hence, if we go through these diagrams, the closest and correct representation of the polar head being outside and non-polar fatty acid tails being protected is option C. Question 18. What are the features of facilitated diffusion? 1. It uses protein channels in the membrane and is driven by the energy from ATP. 2. It moves molecules from regions of higher concentration to lower concentration and is driven by the kinetic energy of molecules which are diffusing. 3. It uses protein channels in the membrane and the maximum rate of diffusion depends on the number of these channels. Facilitated diffusion doesn't require ATP, hence option 1 is obviously incorrect. The answer is 2 and 3. Option D is the correct answer. Question 19. Which description of chromosome structure is correct? A chromosome consists of a long DNA molecule wrapped around histone proteins. Therefore, the answer is D. The very long DNA molecule of each chromatid is coiled and held together by proteins called histones. That is the description of a chromosome structure. Question 20. Which processes involve mitosis? Mitosis is involved in clonal expansion and not selection. Over here they have mentioned clonal selection of T lymphocytes, therefore it cannot be included. Forming cells at the tip of a growing root, yes. Replacing skin cells damaged by an injury, that is also correct because mitosis is involved in cell replacement also. Therefore the answer is C. Question 21. A drug has been developed to treat certain types of cancer. It prevents mitosis by binding to the spindle and preventing sister chromatids being separated and moving to opposite poles of the cell. The photomicrograph shows cells in different phases of mitosis. What represents the stages of mitosis that will be able to occur in a cell which is entering prophase when treated with this drug? So the photomicrograph shows cells in different phases of mitosis. Obviously prophase is the first stage. Separation of sister chromatids occurs at this stage called anaphase, and that process will be prevented. Hence, anaphase doesn't occur as there is no separation of chromatids. Therefore, the answer is A, 1 and 2, because 1 is represented by prophase, 2 is represented by metaphase, 3 is anaphase. Therefore, since anaphase doesn't occur, only 1 and 2 will occur when entering prophase when treated with this drug. The answer is A, 1 and 2. Question 22. Some viruses have single-stranded DNA as their genetic material. This DNA molecule has to be folded to fit in the protein coat. Which statements about single-stranded DNA are correct? 1. Single-stranded DNA cannot replicate semi-conservatively. 2. If only the percentage of cytosine is known, then the percentage of guanine can be calculated, but the percentage of adenine and thymine cannot be calculated. 3. Hydrogen bonds may be present within the DNA molecule. Since they are talking about single-stranded DNA, percentages of bases cannot be determined. Hence 2 is obviously incorrect, as in a single-stranded DNA, base pairing doesn't occur. Therefore, the answer is C, 1 and 3 only, as all the other options contain 2. Question 23. How many statements are true for semi-conservative replication of DNA in a eukaryotic cell? 1. The process takes place in the nucleus. Adenine will line up against uracil on the complementary stand. Each new molecule will contain one strand of the original molecule. 4. If the original molecule contained 40% guanine, each new molecule will contain 20% guanine. In DNA replication, A lines with T, not U, as this is talking about DNA replication. The answer is B. 2 statements are correct, and that is the process occurs in the nucleus and each new molecule will contain one strand of the original molecule. Option 2 and 4 are incorrect. Option 4 is incorrect because if the original molecule contained 40% guanine, then the new molecule will also contain 40% guanine, not half of it. Since only 2 statements are true, the answer is B. Question 24. Part of the nucleotide sequence of an mRNA molecule is shown with spaces between the codons. As you can see, this is the mRNA molecule and the nucleotide sequence. The translation of the codons is provided, which events will cause the termination of polypeptide synthesis during translation. Deletion of C from the LU codon. That will not cause termination as it doesn't lead to UAA anywhere throughout. If you split the nucleotide sequence into 3 bases and then apply the correct amino acid, you won't get the stop codon if 1 is carried out. Therefore, the answer should not contain 1. Hence, the answer is D, 2 and 3 only. Question 25. External factors change the rate of transpiration, which also affects the water potential gradient of cells in the leaf. Which combination is correct? Factors that lead to increase in water potential gradient are increase in light intensity, increase in wind speed, increase in temperature, and decrease in humidity. Therefore, if we go through these options, decrease in temperature doesn't increase the water potential gradient but decrease, hence A is incorrect. Decreasing wind speed. Decreasing wind speed won't increase the water potential gradient as I mentioned in these factors, therefore B is also incorrect. Increasing relative humidity, the water potential gradient decreases. That is correct. Increasing sunlight, water potential gradient decreases. That is incorrect, therefore option D is also incorrect. The correct answer is C. Question 26. The photomicrograph shows a transverse section through a leaf. Which features of a xerophytic leaf are visible in this section? 1. Sunken stomata. 2 layers of epithelium. 3. Thick cuticle. 4. Small surface area to volume. So they are asking about the features that are visible. You cannot visualize a small surface area to volume ratio in this photomicrograph given. Therefore, the option should not contain 4. And you obviously cannot see 2 layers of epithelium. Hence 2 is also incorrect. The answer is C, 1 and 3 only. Question 27. Which changes occur as carbohydrates move out of a root cell into a phloem sieve tube? Now this process represents the loading of sucrose, which leads to water potential gradient being more negative in sieve tube as carbohydrates are moved into the phloem sieve tube. When carbohydrates move into the phloem sieve tube, the water potential gradient decreases. Therefore, the gradient becomes more negative, which leads to an increase in volume. When the water potential decreases in phloem sieve tube, volume increases and vice versa. The answer is option D. Water potential in a phloem sieve tube becomes more negative, as more negative means decrease in water potential. Volume of liquid increases because decrease in water potential means volume increases as water enters the phloem sieve tube when there is a decrease in the water potential gradient. Hence, the answer is D. Question 28. The photomicrograph shows 3 white blood cells labeled X, Y and Z. Which row correctly identifies these cells? The cell with a lobed nucleus is neutrophil. Kidney-shaped nucleus is the monocyte. Circular nucleus is of lymphocyte. X represents lymphocyte. Z represents neutrophil. Y represents monocyte. Hence, the answer is A. Question 29. The graph represents data on blood vessels and blood flow. Which row correctly identifies the curves? So total cross-sectional area is highest in capillaries. Therefore, if you look at these curves, X represents cross-sectional area as it is the highest at capillaries. And speed of flow is the lowest in capillaries. And the lowest curve represented over here is curve Y. Therefore, the answer is C. Speed of blood flow is Y. Pressure of blood is Z. And total cross-sectional area is X, as cross-sectional area is highest in capillaries. Question 30. Which components of blood are present in lymph? White blood cells, proteins, sodium ions. In lymph, white blood cells are present, proteins are present, and sodium ions are present. Hence, option A is the correct answer as all 3 are present. Question 31. What correctly describes the significance of the increase of red blood cell count of humans who move from living at low altitude to high altitude? When moving from low altitude to high altitude, the partial pressure of oxygen decreases. When the partial pressure of oxygen decreases, the RBC count, which is known as the red blood cell count, increases to compensate for the reduced oxygen. Therefore, the answer is A. After a few weeks at high altitude, the number of red blood cells increases to compensate for the lower availability of oxygen. In other words, to compensate for the lower partial pressure of oxygen. Therefore, the answer is A. Question 32. Which structures of the human gas exchange system are supported by cartilage? The key thing to remember over here is that cartilage is only present in trachea and bronchus and not in bronchiole. In the trachea, it is present in the form of C-shaped rings, and in bronchus it is present in the form of irregular blocks or irregular plates of cartilage. Since the bronchiole doesn't have cartilage, the answer is D. Question 33. Carbon monoxide, nicotine and tar are components of tobacco smoke. Which row correctly describes their effect on a smoker's body? Carbon monoxide, it forms carboxyhemoglobin as well as a stable compound with hemoglobin. Nicotine leads to various effects. It increases the blood pressure. It also increases the heart rate. It leads to constriction of blood vessels. And it also makes the platelets sticky. Tar leads to stimulation of goblet cells, releasing more mucus. And it destroys the cilia, lining the airways. Therefore, if we go through these options, for A, the effect of increasing the diameter of blood vessels under nicotine is wrong, as the diameter doesn't increase when nicotine is present but rather decrease. Hence, option A is incorrect. Option B, forms a stable compound with hemoglobin. Yes, that is correct for carbon monoxide. Increases blood pressure. That is also correct for nicotine. Stimulates goblet cells to secrete mucus. That is also correct for tar. Hence, the obvious correct answer is B. If we go to option C and D, option C, carbon monoxide forms carbaminohemoglobin. That is obviously incorrect, as carbon dioxide forms carbaminohemoglobin and not carbon monoxide. If we go to option D, forms carboxyhemoglobin, yes. Decreases the heart rate, that is incorrect, because nicotine leads to increase in heart rate. Therefore, option D is also incorrect. Correct answer is option B. Question 34. Which statements about chronic obstructive pulmonary disease are correct? 1. The disease can often be reversed by treatment. 2. The patient's symptoms normally do not change. 3. The patient is normally over 30 years old. 4. The patient coughs frequently, producing a lot of mucus. COPD is a condition that cannot be reversed by treatment. Hence, option 1 is incorrect. Therefore, the answer is D, 2, 3 and 4, as all the other options contains 1. Question 35. Which of these causative agents of disease can be transmitted by droplet infection? Vibrio. Vibrio leads to cholera. Mycobacterium leads to tuberculosis. And option 3, morbillivirus, leads to measles. And Vibrio, which causes cholera, is waterborne, hence not droplet infection, whereas Mycobacterium and morbillivirus represents droplet infection. Therefore, the answer is D, 2 and 3 only. Question 36. The table compares total antibiotic use and the percentage of antibiotic-resistant pneumonia cases in 5 countries. As you can see, the countries are Austria, Denmark, Iceland, Spain, USA, and they have given the total antibiotic use and the percentage of penicillin-resistant pneumonia cases. They are asking what is supported by the data. If we go through these values and their relation, the answer is B. Option B is represented as when X increases, there is an increase in Y. Question 37. Which statement about the development of resistance to antibiotics in bacteria is correct? A. All mutations in bacteria result in resistance to antibiotics. B. Antibiotics increases the rate of mutation in bacteria. C. Mutations leading to antibiotic resistance only occur when antibiotics are used to treat pathogenic bacteria. D. The proportion of antibiotic resistant bacteria in a population only increases if the antibiotic is used. So option D is correct, as antibiotic resistant bacteria in a population will increase only if the antibiotic is being used. If the antibiotic is not being used, there cannot be any resistance being developed, as no bacteria comes in contact with the antibiotic. Therefore, there cannot be any selection process, and hence the antibiotic resistant bacteria do not occur. Therefore, the answer is D. Question 38. A blood cell count can indicate how many white blood cells there are in the blood. Where else in the body are white blood cells found? 1. Bone marrow, 2. Lymph, 3. Lungs. White blood cells are present throughout the body. Therefore, it is present in all 3 of them: bone marrow, lymph and lungs. Therefore, the answer is A, 1, 2 and 3. Question 39. Myasthenia gravis is a disease that results from the immune system failing to distinguish between self and non-self. Antibodies bind to a component of the junctions between a muscle and its nerve. What correctly describes myasthenia gravis? A. Impulses in the nerve to the affected muscle cannot be blocked. B. The affected muscle cannot be repeatedly stimulated. C. The affected muscle is permanently stimulated. D. The nerve to the affected muscle does not carry any nerve impulses. C is obviously incorrect, as the affected muscle is not permanently stimulated but it is reduced. Option D. The nerve to the affected muscle does not carry any nerve impulses is also incorrect, as it does carry some impulses but it is not enough to stimulate the muscle. Hence the option which is correct is option B. The affected muscle cannot be repeatedly stimulated. Question 40. A monoclonal antibody specific for a virus was treated with an enzyme to break the bonds between the variable and constant regions. The separated variable and constant regions were then added to cells infected with the virus. Which statements are correct? 1. The constant regions would bind to different parts of the virus antigens. That is obviously incorrect, as constant region, it has a specific shape, it binds to a specific receptor. Therefore, statement 1 is incorrect. Hence, we have to consider between options 2 and 3. Option 2, the viruses could be engulfed by phagocytes if they were present. Option 3, the variable regions would all bind to the same part of the virus antigens. Option 3 is the correct answer, as variable regions, they form the antigen binding sites, and antigen binding sites have a specific shape. Since they are specific in nature, they only bind to a specific substance or a specific antigen. Hence the answer is D, 3 only. And that is the end of this paper. Want to solve more unanswered questions? Well, you could click on my next video. Nonetheless, thank you for watching. Appreciate it, and do stay tuned for more.